Hello everyone, my name is Dr. Vera Gonzalez. Um, Basically, my background is bioengineering, and during my PhD, which I finished last year, I was doing a lot of neurotechnology, so basically engineering applied to neuroscience. And then I got this fellowship, and basically I started learning ML and AI because I literally knew zero before I started. And so this talk is going to be a bit of an intersection between neurotechnology, neuroscience, and computer vision. So if you have like questions about the neurotechnology, I probably can answer whatever. If you have questions about the computer vision, for crossing fingers, I know it. And if not, I'll refer you back to the papers. So the first half of this lecture, I'm going to be talking about basically the basics of computer vision. So we're going to see like the main like classical networks, and we're going to try to see like intuitions of like how they work, these networks. And we're going to see them through examples in neuroscience and neurotechnology. Then we're going to have a small break, and then we're going to have a second half that's going to be a bit more practical. So we're actually going to look at like some like very easy code, on how you actually implement. For example, we're going to be looking at like image augmentation and even like a small CNN. And we're going to be doing it through the lens of neuroscience. So this second part, actually, we're going to be talking about a technique that is called patch clamp, which I'm sure sounds like nothingness to you. But I'm hoping like by the end of the lecture, you're like complete expert on patch clamp. Basically because that was mainly what I was doing during my PhD. So it was the automation of this technique. Okay, cool. So I think we all know why you want to study the brain, right? Like it's not only about understanding how it works when it's working properly, like you might be able then to control a robot arm, or you might be able to control like some computer brain interface, but you also want to understand what exactly goes wrong when you have like a disorder like Alzheimer's disease and something like that, right? And there are many ways that you can look at the brain in kind of different levels that you can look at it. So if we start with a top-down approach, you can have imaging modalities that look at the whole brain, right? So that's like CT, for example, or MRI. This here is a functional MRI, which basically allows you to see where the oxygenated blood is within the brain. And so basically that tells you what areas of the brain are the most active. So say that you have someone doing a task, a motor task, or you have a person trying to recall a memory, then for the person doing the motor task, you have the motor cortex being activated, and so you're going to see that in red. And for the person that is trying to recall a memory, you might have, for example, the hippocampus lighting up. So this gives you an idea, this kind of technique gives you an idea of like what areas of the brain are in charge of what functions. Then you can have, a bit going further down, you can look at particular brain regions. And so one of the techniques that are used for this is calcium imaging. So calcium imaging is mostly done in animals, not in persons, like not in people like the other one. In calcium imaging, basically, you have an animal that either is born expressing a gene for a fluorescent protein, or you can inject it with a virus, and the outcome is that the cells are basically going to light up. And the intensity with which the cells are lighting up basically depends on the amount of activity. So basically calcium is just a proxy for how much activity the cells have. And so this kind of images is the thing that you get with calcium imaging. You can have small fields of view that look like this. So you have like, this is a cell, that's a cell, that's a cell. And you have one cell in each of these squares, another cell. So you can look at quite a big amount of cells, or even you can have these kind of images where each, you cannot even see, but like everything, the green are just basically squares. So this is called mesoscopic calcium imaging, just meaning like you have a really big field of view. And so this again, you can have the animal doing a certain task. For example, you know how there are neurons that are called place cells, that basically whenever the animal is going through a particular place in a maze, it will light up. Like that cell always lights up in the moment the animal goes through that particular point in the maze. And so this is the kind of things that you can look at. So you're looking now more about like local regions and how they are working. And finally, you can go even farther down and you can just look like to a few cells. And so that's what targeted electrophysiology and in particular patch clamp deals with. So you basically, this is something that you could be looking at. Again, you have these cells that are expressing a fluorescent protein, in this case it's green. And then you can have basically a pipette with an electrode coming in, and then you can measure the electrical activity of this cell, and maybe you have another pipette in this cell, and now you can see, are they connected? If they are connected, how they are communicating, how do they respond to different stimuli? So there's also a lot of information that you can get from doing these kind of studies, which is this targeted electrophysiology is basically, an example is the patch clamp technique, which is what we're going to see in the second part of the lecture. And you might be thinking, okay, why on earth do I want this if I can have all of that? And the answer is that you don't get as good accuracy and temporal resolution when you're doing a lot of cells at the same time. So the idea here being that as the field of view increases, then your temporal resolution is decreasing. So basically you want to be doing all of it, right? You don't want to be focusing only on one technique. You really need all of them to be able to understand what's going on in the brain. So now going to computer vision, there are many things that you can do, obviously. Some very common tasks, especially in the neuroscience field, are object detection, image classification, image segmentation. You can also have neurostyle transfer, which is basically this. But for example, some examples of object detection would be you're looking at a scan of the brain, and then you want to be able to detect if there's a stroke or if there's a hemorrhage, something like that. There's also a lot of like image segmentation as well. So for example, that's being used in surgeries, like you're able to segment where the tumor inside the brain is, and then that would help the surgeons to kind of remove it. But with all of these techniques, the big issue is that the input to whatever neural network you're working with is that they're very big, right? Images usually want the most quality, which in turn will mean that they are huge. So if you're working with like a normal ML kind of paradigm of like neural networks, if you have like these fully connected layers, you're basically going to have a incredible amount of parameters, but you're going to have millions and billions of parameters to train, and that in turn means that you're going to have a very slow model, and it's going to require a lot of data to be able to work, right? And so obviously that's not something that we want. So the solution for these are convolutions and convolutional layers. So I think everyone knows here what a convolution is, but just in case, the basic idea is that here, for example, you have a 3x3 filter, and you're going to be moving it along until you have gone through the whole image, and then you're basically multiplying each of the values of the filter by the pixels in the image, and that's going to give you a convolved feature. So the intuition for this is, for example, let's look at convolution with an edge detector, which is this. This is what this is. So if you have in an image, obviously you don't have such a small image, but imagine this is like a part of the image, right? And you have an edge, basically. So you have all of this part is one color, and all of this part is another color, right? So that's an edge, a vertical edge. So by applying a convolution with this kind of filter, and there are other types of filters that also do the same thing of edge detection, such as Sobel filter and Shar filter and so on. That's effectively going to tell you, okay, you have an edge right down the middle, and it's a vertical edge. And so in the same way that you can do this to get the vertical edges, you can also rotate it, and then you can detect horizontal edges, and you can flip it and have 45 degrees edges, and so on. And then the basic idea in convolutional neural networks is that you're going to have these convolutional layers that are basically extracting features, right? So a way that you can think about it is, okay, the first few layers of my model are basically going to be detecting just vertical and horizontal and 45 degrees edges. And then the middle layers are maybe getting all of that information together and saying, okay, I have squares here, I have triangles here, I have this other thing going on. And then by the end of the network, so towards the last layers, you're basically getting more information about like more detailed features and like texture and so on. So that's how we think about the convolutional neural networks for images. And very often for example, if you have a classification model, so you basically want to be able to detect in an image, or do you have a person, a car, a bicycle, whatever, right? At the very end, you usually have one layer where you're flattening basically all of your 3D convolution layer. You're going to flatten it all together in one vector. And then usually you will have one or several fully connected layers. And then as your normal ML algorithm, you have a softmax just to determine which class the object is. There's also very common in convolutional neural networks are the pooling layers, which we are gonna we talk about after this. Okay. So in convolutional neural networks, there's basically two main advantages over the fully connected layers or the traditional neural networks. The first one is the sparsity of connection. So basically, when you're doing a convolution, you know that the one pixel on the second layer depends on 9, if your filter was 9 by 9, on 9 pixels of the original one, right? So there's basically a connection of 9 to 1. So it's only as big as your filter. So that's quite sparse if we compare it to a fully connected layer where you basically have everything multiplied by everything. And so obviously this helps with computational, like you're reducing the computational cost. And the second idea is parameter sharing. So the idea here is that if you have a filter that is able to detect edges, for example, in one part of the image, then that's also true for everywhere else in the image, right? So then you're basically, the parameters that you're training for are the 9 numbers in your filter, right? So then those 9 parameters will be useful everywhere in the image. And so again, this also helps you with the lowering the cost of the computational power. And pooling layers are also very, very common in CNNs. Basically, the idea is that you're doing either a max or an average pooling, meaning that you're getting, in this case, out of 4, you're getting the maximum or the average, and that's what comes out. And so a way that you can look at this is that you're basically subsampling. You're retaining only the most important features. And because these layers do not have any parameters that you can learn, then very often in literature you will say that they'll talk about a network that only has 2 layers. But actually each of these 2 layers has convolutional plus pooling layer, and that would be counted as 1, because you're not learning anything in the pooling. Okay, so just a small quiz, just to see if you were listening. I don't know, because I'm supposed to include you right in the lecture, so which would go with what? So what do you think the convolutional layer is doing? Is it doing classification, feature extraction, or subsampling?
Feature extraction.
Yay! Yes. And what is the pooling layer doing?
Subsampling.
Yes. And then the fully connected layer is for classification. Cool. You're listening. Okay, so we start with some famous CNNs. So the Linnet 5 was like one of the first ones to be out there. It was a very simple network. It basically had like 4 layers. So 2 of them were this combination of comb plus pooling, and then the last 2 were fully connected layers. And the Linnet network, basically what it did was that if you gave it a handwritten number, it would tell you what number that handwritten number is, which sounds like so trivial, but actually it's not that trivial for the thing. Then AlexNet, basically, it was quite similar. It just went like bigger is better. So it had more training data and more layers, and that was basically it. Then for the VGG16 and VGG19, these are actually very, very big networks. Even by today's standards, it's considered very big. It has like something like 120 million parameters or something. And this is what it looks like. Again, it's just you're just adding more and more layers. And as you know, the more layers that you add, then the more likely you are to have either vanishing or exploding gradients, which is a big issue. So that's why ResNet came up. So ResNet, or residual network, basically came up with the idea of having this kind of skipped connection. So basically you have your input going through 2 layers, and then given at this point, you add up back the input from before. And this was actually very, very useful because usually, so in theory, you would think that the more layers you add, it's all going to be fine, your training error is going to go down. But actually, because of the vanishing and exploding gradients, the reality is that the training error actually ends up going up if you add too many layers. So ResNet basically got rid of this problem by adding these skipped connections, because it's basically helping the network to learn the identity function, and so it's avoiding that it will lose important features, basically. So this is an example of a ResNet. Basically, if you look at anything of the literature, there's so many ResNets out there, like so many, because obviously, like anything that has this kind of skip connections, you can just call it a ResNet. Yeah, so this is just an example of like how does it go from like the normal network to having this ResNet. And actually an example of a ResNet, or an example of a network that has ResNet as its backbone is RetinaNet. So RetinaNet basically does object detection, and it has this pyramidal structure, which is a feature pyramidal network, they call it. And basically the idea here is that you have feature maps at different sizes. And so then this helps you find objects that have different sizes. So like the smaller objects will be detected here, bigger objects will be detected here. And that's kind of the idea. The other important feature that they brought that was new was the loss function. So they used a variation of the cross-entropy loss function, where basically they were giving an extra term so that you would give more importance to objects or classes that were self-represented. So imagining that you have like cars and people like walking around the streets, most of your image is actually background. And so what this network was trying to do is apply a loss function that will actually help the algorithm detect the people and the cars and the things that are less represented but you're more interested in. Yeah, and so an example of this network being used in calcium imaging is this paper that actually came out like less than 2 years ago. And it was like a very talked about paper in the field because it proposed this pipeline that works really well both online and offline. And basically what it does is you have like a video basically of the cells being fluorescent a long time, which usually corresponds to like an experiment that the mouse is doing, right? Of maybe he's moving a lever or he's like going around a maze or whatever other thing. And so basically the algorithm is gonna first denoise and just does some pre-processing basically to the videos, and then it applies the RetinaNet to detect where the cells are. And so basically you're going to have these bounding boxes that are detecting where the cells are. So then later on you can extract the fluorescence from each of these squares like this, and then you're going to get basically a fluorescence signal that then via the convolution you can transform into the real electrical activity, because you're measuring calcium as a proxy for the electrical activity. So yeah, this was again a very, very powerful pipeline. It works really well, and yeah, it's using the RetinaNet. Okay, another example of a pipeline that is very used in the world of calcium imaging is Cayman. For very long I call this CalmAn. I'm not very sure why, but yeah, it's Cayman. And it's an open source pipeline where basically it's the same idea as before. You have like your videos of the cells doing their thing. So you first do some motion correction, some denoising, then you identify where the cells are, do the mix overlapping signals. So that's important because basically here in this example, like it might not look like much to you, but it's 3 cells, one on top of the other. And so the software is actually able to extract each of these 3 cells and what they are doing. And finally, you deconvolve the fluorescence signal, which is this one, to obtain the actual electrical impulses of the cells. And it uses this constraint non-negative matrix factorization, which sounds very fancy, but what it means is that with all of your data, you're dividing it into two matrices. One that is temporal, so it just stores what is the activity of each of the cells a long time. And the other one is geographical, so meaning it stores where the cells are within the picture. And the idea here is like they're just averaging along a lot of frames, so that even if the cell is not having any fluorescence at one point, but it's having it later, then you're able to detect it and then just check a long time, because obviously the cells do have periods of time where they're doing nothing, they're just chilling. And also one more kind of also related to this. So Deep Lab Cat is basically a software package to estimate animal pose, and you may think, okay, why want that? The idea here being that there's so many things that you can do to basically alter either the genes or some particular, like you can give them an injection of some chemical that basically will deactivate a particular cell type within the mice, or you can give them, they can have a gene alteration, or even like whole brain regions be altered. And so the idea here is that by quantitatively measuring the behavior of the mice, then you know what is the effect of basically that gene or that cell type or that whole brain region. I don't know if that's clear or yeah?
Then you just see how the animal behaves after.
Yeah, so basically, for example, in my lab, they work with Alzheimer's, mice that have Alzheimer's. And so they basically see how they perform in memory tasks as compared to normal mice that don't have the disease. And they also can see a long time how is their decline, right? And so there's many things that you can know then of how the disease progresses and so on. Yeah. Okay, so now the Inception, also known as Google network. So the Google Net is called like that because basically it was obviously developed by Google, and the fact that they have the net here is just like kind of trying to make or honor the original net. And it's also called as the Inception network because this meme is literally cited on the paper because that was the inspiration. We need to go deeper, like the movie says. So meaning they just wanted to have more and more layers, basically. And so how do we do that, right? Because if we add more and more layers, then we have more and more convolutions, and then we're going crazy with the amount of computations we need to do. And the idea that they came up with was the one-by-one convolution. So the idea being that you're gonna convolute with a filter that is just 1 pixel in width and depth. And that's gonna basically allow you to reduce the dimension. And you might think, okay, what's the purpose of this? Well, basically the idea is that you're going to apply this kind of convolution before you apply whatever convolution you wanted to apply from the beginning. So in this way you have basically reduced, because basically convolutions help you reduce both W and H, right, and pooling layers as well, but nothing helps you get rid of D. And so the one-by-one convolution basically allows you to also reduce D. And then once you have this, you can apply whatever convolution you wanted to apply from the beginning, and then that means that you don't need to do as many computations. So
here the width and the height is of the.
So this is basically when you first start, right, on your the input, you will have this would be your image. So you have 1,000 by 1,000 pixels, and maybe you have 3 channels, so you only come up to here, right? So you only have 3. But then as you continue with the layers, you apply several convolutions and several filters within the same layer. So then you're going to be expanding almost constantly on the D front. So then this one-by-one convolution is allowing you to go back to smaller. And so this is an example. Maybe it's a bit like boring because of the math, but basically it shows you like one particular example where basically your computational cost is the number of filter parameters times the filter positions, meaning how many times you move the filter along the image, times the number of filters you're using. And so if you wanted to apply to this kind of image, so 28 by 28 by 192, and you want to convolute it with 32 filters that have this shape. So that's going to give you this. And the number of computations you need to do is like 120 million, so like a really big amount. However, if you do first a one-by-one convolution, you're going to obtain this middle step, and then you perform these 32 filters that you wanted from the beginning of 5 by 5. In this case, the total computational cost is literally a tenth of how much you wanted in. Yeah.
So when you talk about computational cost, it's like the cost of just doing one forward pass, not the cost of training.
No. So just well, to be fair, at the end, it ends up being that overall in your network is going to be much better. But yes, like here we're literally just looking at one layer convolution. That's it. Yeah. Cool. Yeah, and so this is the inception network, or well, to be fair, at this point, for all of the networks I'm talking about, there have been like so many versions of each of them. So I'm mostly talking about like the first one that came out. But as you will see, if you look in the literature, there's so many that have come out afterwards. And so, okay, basically this one, I know it's confusing, but it starts here and ends there, so it's right to left. And as you can see, basically you have a lot of blocks that are exactly the same. And so this is called basically the inception layer, meaning that you basically apply to each of the convolutions that you wanted to apply. You apply first a one-by-one convolution. And in this way, you're basically, because you're reducing the cost, you're able to apply even more stuff. So what they do is that they apply 4 different things to the same input, and then they just concatenate the results. The idea being you're extracting different features for each of these 4 operations, and then you're just concatenating those 4 feature extractions. And so they do this kind of block or layer many times. And then these 3 here are basically the classification branches. And the reason you would think that you would only have it here at the very end, right? Like you just want to classify at the very end. But actually what they were thinking was like, wait, so I want to make sure that like whatever is in the middle, like on the way to get me my final solution, has to make some sense. So then they are adding these classification layers here, so that basically you're making sure that whatever is kind of in the middle of the network is still making sense. It's still somewhere close to what you're going for. Yeah.
And so in the loss, you take into account all 3.
All 3, yes.
So
And I'm guessing not at the same level. To be fair, like I haven't looked it up, but I'm guessing not at the same level. But yes, you want to consider all 3 because you do want to make sure that you're like on the right track. And this is an example where they use the Google Net, and actually in the second part we're also going to look at an example where they use the Google or Inception network. And here basically they just used it to obtain the artifacts that come up when you're doing a CT. And then they would use basically this map of all the artifacts and subtract it from the original image to get these high quality images. And yeah, that's just a very straightforward example of how this can be used. Cool, now we look at mobile nets. So mobile nets are basically the networks that were designed to be run on mobile devices or anything that doesn't have very good computational power. And so again here we're doing something weird with the convolution. So the idea here is that we're going to separate the convolution. So a normal convolution would look like this, right? So you have your filter that you're going to, these being your 3 channels RGB of the image, and you're going to be convoluting it along this, all of these pixels. And if you have D prime filters, then your output is going to look like D prime. And this W prime and H prime basically just come from this sliding through each of the pixels. So that's your basic convolution, right? Now what the depth-wise separable convolution does is that it first multiplies 1 filter per channel that you have and obtains this middle stage, where afterwards by doing a 1-by-1 convolution of D prime filters, then you end up getting the exact same result that you got in the normal convolution, but you're doing it in 2 steps. And once again, this idea of you're separating it into 2 is also going to help you in terms of your computational cost. and in fact, they measured in the paper how much it was that you could save, and basically it depends on d prime, which is basically the final dimension that you want in your output layer, so not the width and the height, but the depth. And dependent also on the filter that you're using. So, yeah, that's what they proved. And actually, mobile nets are very used nowadays because they just don't need as much power as other networks. And yeah, these two were just like the first versions of mobile nets. Here the idea being, so you had these blocks repeated 17 times, so you had 17 of these like separated convolutions, and the idea here being that basically during the time that is you still have the image or layer expanded, you're allowing the model to learn on like the high level like details, but then you project back to smaller thing before you go on to the next block, so that you save memory. And okay, yeah. So the next network that we're going to be looking at is called YOLO. Does anyone know what YOLO stands for in computer vision? Not in the. No one.
I see the model.
You've seen the model,
Yeah.
and you didn't know what it stood for.
No.
Okay. So I mean, I would say it's quite catchy what they did. So it's you only look once. So here the idea being that imagine you have a CNN that has been trained to recognize objects or parts of objects, okay? And so if you had that already trained, imagine, right? That network is done. And now you want to know within a picture where in the picture my object is. And you want to output a bounding box, so a box that encloses that object. So a very easy way of doing it would be to just apply that network that you already have trained and kind of slide it along the picture, right? That would be a way. But that would be very expensive because you're running the network along so many of your pixels, and in fact, very often you'll be looking, depending on how big your filter size is and how much you stride this convolution by, then you might be doing this crazy amount of work that maybe is not necessary. So the idea of the YOLO algorithm is that, okay, we're going to divide the picture into a grid, basically. And I think the original paper used 19 by 19, but really you can tailor that. And the idea is that now we're going to apply the network and only once. So I'm going to apply that network that recognizes my object only once here, once here, once here, and so on. And the idea basically is that you're going to have these labels for each of these grid places. You're going to obtain a label that looks like this. So where you have P, which is there an object or not, yes or no, binary. Then if you do have an object, yes, then you basically are looking here at the B, it's just Bx, By, Bh, and Bw, is just related to the bounding box. So it tells you where it starts, where it ends, and also what's the height and the width of the bounding box. And then C1, C2, and so on until Cn just tells you what classes you're looking at. So, for example, in this example that you're detecting dogs, bicycles, and cars, if that's the only three objects you're looking to identify, then you will have C1, C2, and C3, and that's it. And so you would have, say that, for example, in this part, this grid, right? This grid place only has a bicycle, nothing else. So in that case, you're going to have a 1 here, and then also a 1 if your class is bicycle, and then 0 for the rest. Whereas if you had here, which is a dog, then you have a 0 for the bicycle, a 1 for the dog, and 0s for the rest. So
if you have two objects,
we're going to look at exactly that, yeah. So how do you evaluate how good your algorithm is at detecting the object? Well, basically what you use is intersection over union, or IOU. So the idea here being that, okay, my prediction is this and the ground truth is that. So how good is my prediction? And so you calculate it by dividing the area of overlap divided by the area of union. And so that's going to give you a value between 0 and 1 for this intersection of a union, which is, to be fair, used not only in YOLO. This is used like everywhere where you have object detection. And in general, anything that is above 0.5 will be kept, and anything that is below you're going to discard because you're like, this is too bad of a detection kind of idea. And then the next step that you have in YOLO is the non-match suppression, which is basically that if you have detected the object several times, you're just going to be keeping the bounding box that best describes or best enclosures the car. And you're going to do that by basically keeping the bounding box that has the highest IOU. And so this example is this now exactly is what we're going to look at what happens if you have overlapping objects, right? So what happens if you actually have positive for two objects at the same time? So for that you have anchoring boxes. So anchoring boxes, and please don't confuse anchoring boxes to bounding boxes, it's different. So anchoring boxes is basically the idea that you're going to have two spots that you can fill per each grid cell, or two, or however many you want. But imagine that you have an algorithm where you only want to detect people and cars. So you would define anchoring boxes, one that looks more or less like the shape of a person, so that would look like this kind of vertical rectangle. Whereas if you want to find a car, then you would want more of a shape like that. That's kind of what you expect. And so the idea here being that whenever in a grid cell you have positive for two objects, you're going to assign the car to number 2 and you're going to assign the person to number 1. And so now your labels is going to look like this. So this is assuming you only have two anchoring boxes, which, to be fair, you could have as many as you wanted. It's really your decision. And so basically you're going to be assigning whatever object was detected to the anchoring box whose shape resembles the most that object. And in this example, I'm just saying, okay, I'm kind of like hard coding these shapes, right? I'm hard coding the fact that, okay, for people it's going to look like this, for cars it's going to look like that. But it doesn't have to be, right? You could literally have just an ML algorithm that is going to be calculating what are the anchor, or what are the size of the objects that you want to find. And then you say, okay, so basically it makes sense to have 3 anchoring boxes of these 3 shapes, because that's the most common shape that you have for object. And so that's how you get rid, or that's how you deal with having more than one object per grid. So just as a recap, because there was quite a few slides on the YOLO, the basic idea is you basically divide the image into a grid system. In each grid, you're going to be detecting as many anchoring boxes as you decided to have. So in the previous example, it was 2 anchoring boxes. And so for each of these 2, you're going to have either 0 or 1 for like you have the object or not. Then you're going to get rid of all of the IOUs that are low and just get the highest one. And then you're going to generate your final predictions in the corresponding anchor boxes. So in this particular example, you have, okay, my dog is here, my bike is all of these, the car is here.
Cool. Question.
Yeah.
So you said you take the maximum of the IOU, so we know the underlying truth of the...
Well, when you're training your network, yes, you do, right? Because you always have, like, whenever you have the input for whatever CNN you have labeled or annotated, is what you call them, annotated images. So you basically you have to give the algorithm a ground truth, otherwise it cannot evaluate if it's doing properly or not.
That's in the training.
Yes, exactly. Yeah. Okay. So the unit is an image segmentation network, and basically what it gives you is a per pixel class labels that gives you a segmentation map. So basically you can, again, if we keep with the example of like cars and people and so on, if you see a normal image, or if you see, for example, this image that you're seeing right now, then I will be in one color because I'm a person, then maybe the screen would be another color because that's the screen, ground is another color, a wall is another color, and it really will segment anything that you give it. And the idea of how it does this, and now we're going to look at the architecture of the network, but basically you have, again, residual connections that are going to be skipping from one place to the other. And to be able to do this, you need to make sure that your input and output at 2 different points is the same size. So for that, you use transpose convolutions, which basically look something like this. So instead of having your normal convolution that you're going from like a big image to smaller size, now you're doing the opposite. You're going from small to big. And so this is what the UNet looks like. And basically the idea here is, okay, you have your input image and you're doing several convolutions and going along this route. And then the gray arrows here are basically these skip connections. The idea being that the information that is traveling through here is kind of high-level contextual information, right? You're extracting features and storing that information only. Whereas the skip connections are going to give you high resolution for the spatial stuff, right? It's going to give you like texture. And the UNet, the name comes from the simple fact that it does look like a U. And yeah. So now we go into region-based CNN. These are also all the rage right now. They're being used everywhere. Pretty sure also outside of neuroscience and neurotechnology. The idea is that it's object detection, so kind of like similar to YOLO, but it's instant detection, which is the technical term for saying you're detecting the object several times along the image. And so basically what it does is just it proposes potential object boundaries, so the bounding box, then you use a CNN to classify the object, and then you adjust the boundaries for more accurate localization. And there's also another variation of this RCNN. By the way, also there's fast RCNN, if you see it around there. It's just, again, another variation of RCNN that is just faster. There's also mask RCNN. And mask RCNN is just basically you have your RCNN output you object locations along the image with the bounding boxes. And mask CNN is just adding another branch that is going to give you a mask for the object. So it's basically going to give you the segmentation plus the detection. And so, like for example, here it's going to be able to detect, okay, this is one boat, another boat there, and it will give you a mask of the objects. And an example of mask, actually no, okay, so that's in the second part. In the second part, we're going to see an example of where they use mask RCNN for patch camp stuff. Okay, another type of architecture are vision transformers. I think transformers became very popular with large language models like ChatGPT. The basic idea is that you have these attention layers where you're basically telling the network what part of the image it should be looking at. You also have multi-layer perceptron, which are basically just your standard neural network layers. And then you have positional embeddings. Here the idea being that in CNNs, whenever you have if you're looking at the pixels that are the top left of your image, those pixels, you know they came from the top left pixels of your previous layer, right? Because you're having the convolution like that, so you know they come from the same part of the image, so you're keeping track of where things in the image are. However, transformers don't work like that. They kind of divide the image in different parts and looks at them separately. So you basically need these positional embeddings to be able to tell where in the image that information is coming from. And this is just an example of like the different layers, how it's starting to look at like different objects in the image, and then eventually it settles for whichever it was looking for. And I'm not going to talk about this a lot because I think like Alice talked a lot about transformers, or she told me she would, I don't know. Yes, okay. But I guess the main thing for me would be to kind of compare like when would you use CNN, or when would you use transformers? I think that's the main takeaway, and you can always look more about the network, like if it interests you. The basic idea being CNNs are kind of focusing on small patches at a time because you're doing these convolutions to detect local features, where transformers you have this self-attention to basically decide what is important and what is not. And so transformers are very good at understanding the global context, where CNNs are not. So basically, if you have very complex images with a lot going on and understanding the global context of the image is important, then you want to use a transformer. However, if you're more interested at local features like object detection or something like this, you probably want to use a CNN. And also the good thing about CNN is that you require less training data. And but transformers, to be fair, which can be computationally intensive, they can offset this because they're able to parallel process. So it's not always true that they are more computationally expensive as compared to CNNs. It will depend basically on the application. Okay, I think we have like a couple more, and then we are finished with the first. I know it's like a lot of networks, but it's basically like a summary of everything that's going on. So, okay, foundational models. Foundational models are kind of, they're also called generalists. The basic idea is that you feed it anything that you want. So it's going to be trained on very large and very diverse data sets, and it's training through self-supervision. So in the case of generalist medical AI, so for medical applications, they're basically, for example, going to give it like different imaging modalities like CT, MRI, whatever. Then you're also maybe going to give it signals like an electrocorticogram or whatever. You're also going to give it genomics, literature like books and stuff. Like you give it anything that you can. And so the output that it's going to give you depends on what they're trying to do. But in theory, you can get anything that you want, right? So chatbot to talk to the patients to see like solve their doubts, or it can help you like create a report on like a particular imaging, or it can help you support bedside decisions. So it really, in theory, can do so many things. I think there's a lot of talk of like all of the things that it could be doing. And so in general, they are multimodal, so they have a broad understanding of many different data types. And after initial training, they can be fine-tuned with smaller task-specific data sets. And some examples, if you want to look more into foundational models, so these are two papers that are basically talking about like what's the latest stuff on generally medical AI. And so these foundational models are very often very good at either one-shot or few-shot learning, which is basically what we're going to look at. The basic idea of zero-shot is that you can recognize that the network will be able to recognize or classify unseen objects, so something that has never seen before, through inference and knowledge transfer. Then one-shot would be like you give it one example of what you want it to do, and then it infers from there. And few-shot would be more in between the zero-shot and traditional supervised learning. And they're especially interesting for neuroscience and in general biomedical applications, because it can help you recognize new or rare phenomena or conditions that were not part of the training data, which is often the case with biomedical stuff. And also, it can help you with retail AI. So retail AI is the idea that many of the networks that are being created at hospitals, they have only been trained in the data of from that hospital, and even sometimes for only one scanner. And because of privacy, it's hard to like share those images. And so then the issue is that those networks are then going to fail when they look at images from a different scanner or a different hospital. So these kind of networks can really help with this idea of like retail AI. And so just an example is MedFlamingo that came out like very recently. It's a few-shot learner, and basically here they were testing how good it did in different tasks compared to the best prior specialist model. So as you can see, it does all kinds of things like generation of reports, answer questions about a picture, classify different pictures, answer medical questions. And this is just like an example of like, okay, you're giving this text of like what has happened to the patient, and you have here a CT of the chest. And so MedFlamingo is saying, okay, there is a blunt trauma to the left lung with a hemothorax, because it's taking into consideration both what happened to the patient and what it looks like on the image. These kind of networks or algorithms raises some ethical dilemmas, right? Because maybe the patients don't want an AI algorithm to be deciding if they get something or not. So something that doesn't have this ethical dilemma is other stuff that is being done in computer vision in neuroscience. I'm not really going to talk much about it, but if you're interested, I recommend you just take a quick Google search, because it is very interesting stuff what it's being done. So it's more about the idea of not I have, like previously we have basically seen, okay, I have all of these images, what do I do with them? This is looking more at what do you do while you are taking those images. And so the idea is that, like, okay, you can try to remove artifacts or improve the image quality while it's being taken. And so, or for example, you can give a low dose to a patient and then try to obtain what the image, or try to infer what the image would look like if you had given it a higher dosage by avoiding to give it the higher dosage. And so obviously these things are very important because anything that we can do to like reduce radiation, reduce contrast, basically is what you give for like certain types of imaging. And usually these are chemicals that are not super good for your body. So again, if you can reduce the dose, then that's great. And again, also if you can shorten the duration of the imaging studies, that's great. There's also a lot of things done in this area with calcium imaging. So for example, I haven't talked about voltage imaging. The idea is similar to calcium, but looking at the voltage directly. The reason why way less people use this is because the image quality is so bad. Calcium image is much better. So now they're saying, okay, well maybe we can use certain AI methods to get better image from voltage imaging, and then that would be inherently be better because it's no longer using calcium as a proxy for the electrical activity. You're measuring the electrical activity directly, and so that would be preferable if we can actually get something to improve the image quality. And here also there's a lot of talks about like how can you denoise all of your images from calcium imaging. Okay, so that was all. I know it was a lot of like networks and stuff going on. Hopefully it gave you like a summary of like what's going on in the computer vision world, especially like neuroscience stuff. After the break, we're gonna have like more practical stuff, like looking at some basic code, and we're gonna also learn about the wonderful world. Small mini tutorial of how you usually do data augmentation for images. So the idea here being you can do all kinds of things with the images. That does not mean that you want to do everything, because sometimes it will make sense to do something and some other times it won't. And so you can do all kinds of things, right? Rotation, translation, shearing, injecting noise. And the idea is that you want to reduce the annotation cost. So this is very important in computer vision because basically having images that are labeled means that someone has sat down and started saying, here is the thing, here is the thing. And that obviously takes a lot of time. So as much image augmentation that you can do, the better, because then that basically reduces a lot your annotation cost. And obviously, you're also going to have increased algorithm robustness because you can improve your feature detection, reduce sensitivity to noise, avoid data imbalance. So say like I have a lot of pictures where my pipette is always at the bottom left corner, then I flip it and now I have a more balanced data set right before I start, which is important, right? Because your network is going to perform much better if you have a balanced data set. And so overall, this is going to perform better to unseen data. Obviously, we don't want to go too hard on the augmentation because then you might overfit your model to the augmented images, which is also not ideal. And you don't want too little because then you don't have enough diversity for effective training. So these are the three most common libraries that you can use in Python for image augmentation. ImGAU, Keras, and Augmentations, which is the one I'm using. I'm not going to say much about ImGAU and the Keras one. Basically, they are very powerful and flexible. It supports a very wide range of image transformation, and it's obviously compatible with like TensorFlow, PyTorch, OpenCV, everything they want. And as you can see, it's like a very easy thing to do. Like you just import it first, and then here you're basically defining, okay, I wanna, in each picture, I wanna be applying these three transformations. So I'm gonna flip the image, I'm gonna crop it, and I'm gonna introduce some Gaussian blur. And so these numbers here, 0.5, 0.1, 0.5, basically tell you the probability of applying that transformation, because you don't want all of your images to go through the exact same pipeline. So you want all of them to be looking different. So that's why you add these kind of probabilities. And so this line here is literally the line where you're applying to whatever image you have, these three transformations, and that gives you an augmented image. The Keras image data generator, if you're already using Keras for like the rest of your model, then it would make a lot of sense to keep using it. It's very simple to use. I mean, the idea here is basically the same, right? You're giving it different transformations, but it's not as extensive as the other two. But if what you're trying to do is simple, then probably that's good enough. And then you have augmentations, again, also compatible with TensorFlow and PyTorch, and it's fast and flexible, and is suitable for real-time data augmentation. So that's actually important because a very typical thing that you would do is that you basically want to get the images that you have, transform them, and pass it through one epoch of your training, and then transform them differently, then run it again. Basically, you're giving it a new set of transformations every time you run, because in that way you're kind of like your network is seeing different images every time you run it. And so that's a very effective way of doing your image augmentation. And so that's why it's important that it's a fast thing to do. So again, we're gonna see an example. So here I'm basically trying out like different transformations that you can do. I'm basically just choosing one of these transformations per image, because I just want to basically see the effects, right? Because you don't want to, like the first thing that you want to do if you want to do image augmentation is you want to see what is the effect of the different transformations on your images, because you might think that something makes sense, and then you look at it and you're like, actually, this does not make sense. And you're going to see some examples. So again, for example, we're changing brightness and contrast with a probability of 1 just because I want to see the effect. And here you're telling it how much you can change the contrast and brightness, introducing some blur, changing the hue saturation values, equalizing the histogram. Here this is introducing basically different types of noise, so Gaussian noise, fog, snow. Basically you're going to see now what it looks like. Okay, this image quality is again not amazing, much better in the computer, but so basically you have, this is your original image, and I have these 4 pipettes, and these X's are basically notating where I have the coordinates for each of the tips. I'm hoping you do see. And then I have a horizontal flip, vertical flip. Here, for example, these are different types of noises that you can add. So random snow, random fog. That basically it just looks like, I don't know, these big circles. Some Gaussian noise. You can also resize, like just crop the image and resize it, giving some blur. This cutout thing, actually, I thought it was very interesting. I hadn't heard about it before. The basic idea of cutout is, I don't know if you can see, there's basically like some like patches of pixels just missing here. And actually the idea of this is exactly the same as regularization. Like you're dropping certain parts of the picture because you don't want your network to become too reliant on certain features. And so the idea really is the same as dropout, right? And yeah, so there's like all kinds of things that you can do. But which transformation, yeah?
Well, actually, maybe this question makes more sense once you're done.
Okay. So which transformations make sense? So for example, for my algorithm, I do want to be able to tell that this is pipette number 1, this is number 2, this is number 3, and this is number 4. So that is important for me. So then that means that these horizontal flipping or rotations do not make sense for me, because my pipette number 1 is always coming in this angle. So that's actually something that my network could and should learn about. However, for example, noise is something that would make sense as long as it's noise that looks similar to something that I could have in my system, right? So, for example, this random fog makes 0 sense for me. I would never have this kind of noise in my system. However, I could have this kind of noise. So then this is something that I probably want to implement within my image augmentation. And yeah, I think maybe that's it for this slide. Yeah.
And so here you do this data augmentation for the training, but I'm wondering, like, sometimes when you add noise, like at least visually for me, like it's easier to see the pipette tip in like this equalized transformation.
Well, to be fair, this is like equalizing the histogram, so you are expected to see it better.
Okay, so yeah, I'm wondering if like this would not be useful as like a pre-processing step as an input.
Yes, exactly. That's a very good point. So yes, for example, I'm not using equalization because I know I'm not equalizing my input images to the model. If I was able to basically equalize all of my images before I feed them to my model, then that's fine. I can use this. But otherwise, yeah, I'm not gonna use it because it's exactly what you mean. Like this is too good of an image. I don't get this.
And why can't you
on live? Because basically you're like live imaging the location of the pipettes, and the system that we're using to do this live feed is already slow. So if basically if we add, I mean we could, but if we add more stuff, it's gonna make it more slow. And I think I'd rather just design a network that is able to detect them without having to pre-process my feed that is already happening. Okay, cool. So yeah, this is just an example of like, okay, I can apply 3 augmentations per image, and then I'm basically trying the transformations that I decided make the most sense for me. So a crop for me is fine. I'm not doing any scaling, but I am shifting, so like just moving the whole thing sideways, and I'm also not doing any rotations because, as I said, that doesn't make sense for me. Another thing important, yeah, so I haven't talked about this. So the key point parameters. So basically, the way I labeled my images are with the X, Y coordinates of each of the pipette tips. So basically, I need to feed this into the argumentations transformation so that the transformation itself keeps track of where the tips are in the new image. So as you saw, like in the previous thing, it was flipping or like resizing things or rotating, and the tips are still correctly localized, right? So basically, that's what this part here is doing. It's just telling the code like, those are my key points. Please transform them at the same time that you transform the image. And so, for example, this is like the first batch that I was looking at. And for me, it did make sense, most of the transformations I was doing. Perhaps this is going too hard on like contrast and brightness change because like you cannot see much anymore. And then, for example, also it gave me some information about like the padding that is happening here, because here what happened is that it shifted the image upwards. And so to fill in the stuff that was like below, it basically was giving me a mirror image, which actually would not be good for me because then I theoretically have another pipette right here, and I don't want that. So basically I changed the padding that I use in my shifting. So most of the image augmentation that you're going to do is just playing around with looking at your images, seeing what makes sense and what doesn't. And okay, I'm not going to show my model because it's like much bigger, but basically this is a very like simple example that you could use for a class classification example using Keras functional API. So basically Keras has 2 types of API, functional and sequential. So sequential is like a simpler one, and it just allows you to like give it different layers and it just go in a sequential manner as the name implies. Whereas the functional basically will allow you to also do residual or skipped connections basically. And as you can see, it's like super easy really to code this little network, because basically you're saying, okay, my convolutional model is going to look like this. It's going to have these layers, that is, I'm going to have a convolution, then a ReLU, then a max pooling, another convolution, another ReLU, another max pooling, then flattening my my input, and I have a fully connected layer here with the activation softmax to basically make the prediction of the class. And then here I'm basically just feeding this that I have defined here, this model, what is the input of my images, which to be fair, I didn't even need to do that. Then you just compile your model, so you have to tell it what optimizer you want to use. In this case, Adam is like very typical. The loss cross entropy would be also very typical. And what metrics do you want to use to measure how good your algorithm is, in this case accuracy. This line here, conv model summary, basically is going to tell you, okay, so the layers that you have are these, and this is the input size, this is the output size. Just basically to make sense, to make sure that like everything is making sense the way you thought. And then these 3 lines basically is all you need to actually train the model. So these 2 are just basically to tell it that you're going to have batches of 64, of size 64, and then you're defining both your trained dataset and your testing dataset. And so here in this line, you're just basically training the model. It's given like 100 inputs, which obviously is very little, but it's just as an example. And then history here, this parameter will basically store all of the loss and all of the accuracy values for each of the epochs. And then you're able to basically plot that and see how your loss is hopefully became. So just an example. So this would be one way to go about the problem, right? To just code your own network. If it's an easy problem, then this makes a lot of sense, right? It's nothing too complicated. However, if what you're trying to do is something more complicated, then obviously what you want to do is transfer learning, right? So the idea behind transfer learning is that you're just going to choose, if you're lucky enough, there will be some architecture that has already been used in a similar problem, in which case you choose that architecture because you have more chances of it working. Otherwise, you can just choose by intuition of what you think maybe will make sense. And very often you actually will need to try more than one. In fact, we're going to look at an example where they basically tried 4 different architectures just to see which one would be better. And so then the idea is that you download the code and the weight of the values of the weights. And then there's like several things that you could do. You could either just freeze most of your layers and just train the last few layers to adapt to whatever classification problem or whatever thing that you're trying to do. Or you can just download the pre-trained weights and then use that as the initialization to train all over again. So yeah, and this is an example of transfer learning where they were basically trying to do, okay, so I was going to say the same thing as me, but not really. So they are trying to find one pipette and they're using bright field microscopy. So this is a type of microscopy that basically works only if you have a semi-transparent sample. So basically, you can use this if you have like brain slices, but you cannot use this type of microscopy if you have anything in vivo, basically. And so, as you can see, I think the pipettes, they are a bit more visible, I would say, than in my images. I think, to be fair, because also I've been showing the best images that I have of the pipettes, but I also have some where it's very hard, like if you don't have a trained eye to where the pipettes are, it's very hard to tell where they are. But yeah, this is, I think, a bit better defined, these pipettes here, than in my problem. And basically, I think they went a bit hardcore, so they went with the ResNet 101. So that means that there's 101 layers of ResNet. And well, to be fair, they got very good results. They compared it to the previous standard way of finding pipettes automatically, which was just like a cross-correlation kind of algorithm. It was not AI or anything like this. And yeah, they got very good results. And now we're going to see another example where they were also automating the whole system of like doing patch clamp. However, they were again just doing it in vitro with this kind of bright field microscopy. So basically, they were just patching cells in brain slices. They cannot do at any point, because of the type of microscopy they use, they cannot do at any point in vivo stuff. And obviously, you're limited if you're only doing brain slices, because you don't have like the whole circuit to know if the answer of the cell is like realistic or not. That's kind of the idea. And so these guys also did the pipette detection, which we're gonna see in the next slide. But this is an example of where they actually tried like 4 different networks to see the performance in finding the cells. And so in the first one, DetectNet, it's basically based on, I forgot which one it was based on. Give me a second. Oh yeah, so it used the Inception network actually. So yeah, the DetectNet basically was like an expansion on the Inception network. So basically the backbone was Inception and then they added other stuff. And they said that actually this DetectNet, even though it had very bad scores, which I will explain now, it was actually the fastest. So this faster CNN, despite the name, if you remember, this is what we looked at before, this region-based CNN. This was the best one in terms of like precision and recall, but actually it was also very slow. This took 0.9 seconds, and this took 0.1 seconds. Which, if you're like live working with like a whole system of everything is moving and blah blah blah, well, you don't really want to be waiting 1 full second for your algorithm to tell you the cell is here. And so here the parameters basically mean, so the precision is they accepted the detection of the cell as good as long as it was within 5 micrometers of the original center. Then the recall just basically means how many true positives in the dataset they were correctly predicted. So meaning, if you have 100 cells here, this DetectNet was detecting 60% or 61% of the cells, kind of thing. And then the F1 score is just an amalgamation of both the precision and the recall. And so, yeah, and they also compared to this other ResNet, because actually the faster RCNN also has a ResNet as the backbone. So as you will start seeing a lot of the new networks that come out, it's really, okay, you have the backbone of this previous network that works, but we added this other new stuff, and now it can do this as well. And this is a very common thing. And then you have this darknet that was basically a version of YOLO. And yeah, so this was the precision, the F1 score precision and recall that they got. And as I said, even though this had the best F1 score overall, that's not the one they went for. They went for this one because that was the fastest. And I think this is literally the last slide. So this is what they did for the pipette detection. It's so extremely easy compared to like the first one we saw, because literally what they did was just like a really small ML algorithm to fit rectangles to the dark side of the images, which in these images were basically just the edges of the pipette. And then they would basically be like, okay, so in my rectangle here is, in my other one is here, then that's where my pipette is. That's as simple as that. And so you might think, okay, maybe they went too hard with the simpleness, because as soon as you have like other stuff in the image, then maybe your algorithm is not going to work so well. But on the other hand, I'm very sure this would be super fast as compared to the ResNet 101 that the other people use, right? So, you decide what is important for you and you go for that. Also, that was also the conclusions. Basically, we saw that there were many different ways in which you can look at the brain, like different scales, and they are all important, and they all do their part in helping us understand. There are many different networks and exciting developments happening like literally every day. Like many of the papers of the applications I showed were like from a few years, going back like 2-3 years, but actually there's so many out there. And then on the second part, basically that you can either choose to build your own network or do transfer knowledge. It's really up to your task and what data you have. And also we looked at some practical implementation of actually how you do computer vision at this very low level of like when you want to do simple stuff. So yeah, that was all. Now talking about the posters, if you decide to do a poster on this talk, I want to give you basically 2 options. So the first one is just choose one of like these very famous networks and just see how it has been applied to your field. Because I feel like not everyone is going to be an expert on neuroscience, so you don't need to do it our neuroscience. You can just choose one network that you're like interested about, or I don't know, or I really like the YOLO, so I just want to look at how it has been applied in my field. And so that would be one idea. The other idea is that you actually just stick to the neurotechnology and neuroscience thing, and you just look at more stuff that is out there, because really there is so much. I really have talked like at 10% of everything that is being done. So yeah, that was all. If you have any questions, and thank you.